In this tutorial, we shall review the maps and tables that are included in the physiology category. You see here a large range of processes, and they are arranged by alphabetical order, so in order to find what you're looking for, you have to browse along them. So let's go to parasite cell membrane, and here we press on transporter of the plasma membrane, and we see many transporters which are embedded in this membrane. In green here you have carriers, in magenta you have antiporters, which are linked to proton transport and the ATP-ADP transporter. In red you have the different pumps, and low here you have the different channels, and where the transport of proton is associated with the co-transport of sodium or amino acid. Here we have a table of all the gene encoding for transport proteins. This table is also connected to the transport database. And here, for example, if we press on this link, we see a lot of information about the small conductant mechanosensitive ion channel. Here we have the family of sugar transporter, once again with a lot of information about the different transporters, importers, and so on. Now we go to intracellular traffic, and you see here also many interesting maps. For example, if we go to the COP2 mediated vesicular transport, we see the formation of the coat, the formation of the vesicle, and eventually the export of the coated vesicle. Here we see Rab proteins are very important in intracellular traffic. We see here a map that describes all the different Rabs in the individual action at different compartment of the infected cell. And here we have a map describing the tethering complex and the various forms of transport from the nucleus to the Golgi, and from the Golgi to the endosomal compartment and back. In this map on the Rab cycle, we see the Rabs and what it undergoes when it participates in binding to the effector. And here the very important T snares and V snares, which catalyze the fusion of endocytic vesicle with the membrane to release their content. Here we see the different maps and tables which deal with protein export. The parasite is known to export many of its protein into the host cell and to the membrane and beyond. And we see here, for example, the subcellular location of exported protein in different compartments of the infected cell. We see here the participation of clip proteases in the export or in the apicoplast mitochondrion and across the plasma membrane. And here a large table compiling all the gene that encode for protein traffic related protein. This is a table that summarizes the list of all proteins which are exported from the parasite, compiled from various sources, once again coded by different colors. And it's amazing that so many proteins are exported. It seems as if nothing is left for the parasite itself to thrive on. We shall now look into hemoglobin digestion and food vacuole. We see here a map that describes the complex process of hemoglobin digestion, which starts with the endocytic uptake of host cell cytosol in form of vesicles, and then the vesicle open up and the hemoglobin is acted upon by various proteases which yield large peptide, and then another group of proteases to provide the large peptide, a small peptide, and then the dipeptide, which exit the food vacuole and are acted upon by specific aminopeptidases. The heme, which is produced here, is polymerized into hemozoin mediated by lipids and heme detoxification protein. Here we see a map which describes the polymerization or mineralization of ferroportoporphyrin X, or heme in other words, which is mediated by several of the resident proteases and heme detoxification protein to produce hemozoin. And in vivo, in aqueous solution, we can imitate this process and eventually end up with beta hematine crystal, which are visualized here in electron microscope. This process is inhibited by several of the very known antimalarial drugs, as is seen here: amodiaquine, quinine, quinidine, chloroquine, and so on. Now we shall move to a picoplast function, and here you see also several maps. One of them is about synthesis of sulfur iron containing protein inside the apicoplast, and another one showed the fatty acid synthesis of the apicoplast, which we have seen before, but all the yellow background are enzymatic reaction taking place inside the apicoplast. Of the apicoplast, this is a scheme taken from the literature which describe all the different metabolic activities that take place in the apicoplast. Now we go to mitochondrial function, and we see here, for example, the biogenesis of cytochrome oxidase, which is encoded mostly by the proteins are encoded by the nuclear genome, and they are imported into the intermembrane space of the mitochondrion, where they are assembled into the cytochrome C, and then we look at the mitochondrial electron flow with cytochrome C is participating, and you see all the path of electrons going from the different components and reaching eventually the cytochrome C oxidase. Although this electron flow is not producing ATP by oxidative phosphorylation, it is very important to preserve the transmembrane proton gradient and electrical potential. Another very important function of the mitochondria is the mitochondrial TCA cycle. Here you see all the cycle, which, unlike in most other organisms, including human, is not fed by pyruvate, but mostly by glutamate to provide the substrate for the electron transport. Now we go to processes which are common to the mitochondrion and the apicoplast, and we see here, for example, the compartmentalization of porphyrin biosynthesis, which starts in the mitochondrion, goes to the apicoplast, and then return back to the mitochondrion for the we shall now go to other functional processes. You see here, for example, the regulation of pH inside the parasite, and this is the parasite plasma membrane with all the relevant transporters, whose goal is to essentially keep an intracellular pH of 7.3, and as a result of all the proton export and so on, we see here that in the parasitophorous space we have a pH of 6.8. And here is another example how the calcium is maintained in the infected cell. Essentially, we see a group of intracellular organelles with the relevant pumps that essentially accumulate calcium and keep the cytosolic calcium at the desired low concentration. Going to other organelles, we find here the table that summarizes the nuclear proteome, and you will see a very long list of proteins that have been associated with the nucleus, and they are once again organized by functional categories: DNA and RNA metabolism, and later on translation, and so on and so forth. It's rather perplexing that so many proteins which are not suspected to be inside the nucleus are indeed found there.